Howdy, my name is Rebecca White, and I'm the CEO and CTO of Pebble Labs. And before I was a scientist, I was actually a farmer, or at least I grew up on a farm that grew cotton, cattle, and peanuts. And I remember so clearly the year that our community planted BT cotton and walking with my grandfather in fields of regular and BT cotton and pulling bowls and comparing and looking at the difference and the wonder and amazement that he expressed at that difference. I experienced firsthand the impact that biotechnology can have on crops and, more importantly, on farmers' lives. And the team at Pebble Labs is working to bring new biotechnology applications to farmers to ensure their success in the fight against diseases and pests. At Pebble Labs, we are harnessing naturally occurring bacteria to deliver the power of RNA biology against urgent crop threats. Our work is focused on global food production and centered around SDG Target Goal 2.4, which is climate resilient agricultural practices. As everybody on this call knows, feeding 2 billion more people with less land and fewer inputs is going to require at least 50% more production per acre. Climate change is making that much more difficult for a variety of reasons. And globally, the losses already to diseases and pests are estimated to be up to 40% per year, depending on the crop. And this is getting worse again with climate change. So we are focused on these urgent threats to food production. At Pebble, we leverage unique expertise in computational biology and bioinformatics, microbiology and molecular biology, agronomy, host production and pathology, and RNAi defense pathways. Our platform is delivery focused with stackable solutions. Again, we're using naturally occurring microbes from crops of interest. And because we're using bacteria from microbiomes of the crops themselves, this can either be a plant or an animal, to deliver the power of RNA for solutions, again, that target diseases and pests. Our microbes are very precisely engineered to deliver specific RNA molecules that have high target efficacy and extremely low chances of unintended impacts. Everybody knows about RNA these days, especially given the last 2+ years with COVID, because the COVID vaccines are an example of using RNA to fight a disease, in this case, in humans and with a vaccine. And this type of mRNA biology is doable in a human because we have antibody based immune systems. But plants and certain animals don't have antibody based immune systems. So we cannot vaccinate them like we can a human. Instead, we take advantage of a natural defense mechanism called RNAi, or RNA interference technology. This is post-transcriptional gene silencing. It's using a non-coding RNA to stop the synthesis of proteins and therefore stop a pathogen or pest in its tracks. RNAi is very effective, but the delivery is difficult. It's a diverse platform. The mechanism itself is conserved in plants and animals and fungi. RNAi has been shown to be efficacious in a lab setting against a variety of pests, everything from fungus to viral pathogens, from insect pests to nematodes. And the ag industry has been trying for a very long time to deliver RNAi exogenously for crop protection, but has been largely unsuccessful. As a result, it seems that the industry is moving towards a GMO model for introducing RNAi to crops. This has inherent problems. So, what are we doing differently? Well, your microbiome talks to you all the time. So, a plant's microbiome talks to it all the time. And by talking to, what I mean is it's delivering molecules, whether they're RNA molecules or protein molecules or lipid molecules or even small chemicals to the host that it lives in. And so, what we're doing with directed biotics is taking that microbiology, we isolate, select, and characterize naturally occurring microbes from the crops of interest, and we combine that with computational biology. So, we design very carefully the molecules that we're producing, how we put them into the microbes, and we do a significant amount of computational biology and bioinformatics screening to look for off target effects in both the host, in the environment, and tier 1 organisms. We put those two things together with molecular biology so that the microbe that we have selected and characterized is now expressing the molecules that we have very carefully designed. And the result is something that we call directed biotics. It is a platform where microbes are continuously and systemically producing RNA from within the host. It's highly targeted and it is easy application to crops for farmers because in the end, again, we are focused on helping farmers win the fight against diseases and pests. And so there are benefits for both farmers and consumers with our platform. We are using biotechnology without creating a GMO crop. We're using the beauty of the host microbe interaction to create stable production throughout a crop cycle so that you get season long protection using our platform via RNAi. The horizon of 2030 is a very short timeline. We really have to make big impacts in crop protection for diseases and pests in particular along this timeline. Our first ag products are targeting potato virus Y and tomato spotted wilt virus, and our platform is ready for new partners and new challenges. We are currently in a fundraising round, Series A fundraising round. So if anyone is interested, please do reach out. I would love to talk to you more. And with that, I will turn it over to my colleague, Stephanie Wedrick, to take any questions that you have. Stephanie is Pebble's Vice President of Development. Thank you so much for your time.